Okay, so yeah, good afternoon, everyone. So we have Professor Meiyappan with us today. So I'll quickly introduce him to you all. So Dr. Meiyappan Lakshmanan is an assistant professor at the biotechnology department here at IIT Madras. So he's the principal investigator in the systems biotechnology and cellular engineering lab. So he's also a part of various interdisciplinary centers in the institute. So for example, one is RBC DSI, some of you may be knowing, and the other one is IBSC. So prior to joining IITM, so he completed his PhD from NUS Singapore. Subsequently, he worked at the Bioprocessing Technology Institute, part of A Star Singapore. So thank you, sir, so we're glad to have you here. Kindly take over.
Thanks, Karthik, for the kind introduction. Good afternoon, everyone. Glad to have you here. So before we begin with the technical aspects of this presentation, I wish to have some sense of the audience. So can I request you to have a show of hands? Those who have a knowledge on biology at the post your high school level. So your degree could be on biology, or you have been exposed to biology in your college program and so on. Okay, so it's quite little. It's fine. And I also want to know, like, how many of you are familiar with Python and have worked with Google Colab? That's good. So because we would be doing a hands-on workshop today later with how to analyze the gene expression data with Python, particularly using Google Colab. So you will be able to follow my TAs as they do along the exercises, so that you can also experience how it can be done. Okay, so without further ado, let's begin. So just a quick dive deep into what is the central dogma of biology, what we call, which is nothing but each and every living organism in this world, they go through this process. So for example, for every human, we look different and we perform certain activities. The other animals, they do perform certain activities. We are distinguished from them. All these is because of the differences in the genetic material, which is encoded in your DNA. So that comes at the top layer in your the organization of your information, what has to be done and how to react and so on. And then this information cannot be extracted. I mean, this has to be extracted through a sequence of processes. So this is a double-stranded molecule, meaning like it has a repetition. You can assume as binary digits like 1, 0, and maybe you can add 2 more digits like 2 and 3. So it will be usually based on nucleotides, and it will be linearly arranged. So the different combinations of these nucleotides have a meaning. And when they do encode a functionally relevant molecule, it is called as gene. And how this is, so each time we cannot keep taking out this DNA. So this will be kept intact. So there is a complex machinery which gets attached to it and understands what is the sequence of information. It reads through, like how your typewriter just reads through or your printer reads through, and then it prints a copy of that molecule. So on both sides, you have the different strands, the nucleotide strand, and one of that will be templated. It will be copied, and this process is called as transcription. And once you are able to get the exact copy of that, and this is a DNA molecule, and this we call it as RNA molecule, which is the riboxynucleic acid and deoxyribonucleic acid. So once this happens, in complex eukaryotes like human or mouse, there is a process which happens, which is for the sake of understanding what you can say is This RNA molecules are unstable. So to keep them stable within the intracellular environment, it adds some simple elements at the tail and at the head. Then this RNA molecule within the cell, all these processes happen inside the nucleus. So then this RNA, it moves out of the nucleus, and where based on the information in the nucleic acids, the different amino acids get assembled and it will form a protein. The assembly of different amino acids into a form is called as proteins, and usually all the activities in any living organism is actually carried out through a protein. So the simple movement of your arms, your thought process and everything, it's like a movement of various proteins and the assembly and activation of those together. Okay, so this linear flow of information is what is underlying as a basic for every organism. And what we are going to focus on is just on this particular process, the transcription. So among the existing DNA, what are the genes which are get copied, which is the RNA is getting synthesized. So why we do this? Say you may assume two different conditions in the normal and in the disease conditions. So a disease condition happens because of there is a difference from the normal state. And we first, as biologists, we first can start tracking it. So everybody has a similar set of genetic material, right? So why this is happening? It could be because of a misregulation in your genetic architecture. So the first thing we may do is to look at the transcription which is happening, whether the same materials are getting copied and the RNA is being generated or not. The transcription is similar or not. If it is dissimilar, how dissimilar it is. So we can start our analysis to understand about the disease. Why, what is the root cause of the disease and so on. So, there are several methods available to study transcription, the information passage from DNA to RNA. At the simpler level, you can do something like a fish analysis, where you just simply hybridize your RNA molecule and then you try to understand what are the actively transcribed RNA. So for simplicity purposes, I will not go into the technical details of this. The main difference is this analysis is very simple and it can catalog only 1 to 10 amount of RNA. Whereas nanostring is little bit more complicated. It can track up to hundreds of genes. Similarly, RT-PCR can also track about hundreds of genes. Then the next generation was microarray, where you just have a plate which is filled with all the known genes and the RNA sequences of that. And then you extract the RNA from the sample and then you try to hybridize with it. Whatever matches, it will elicit a light signal. So based on the intensity, you will know the amount of that particular gene being expressed. So I will be using a term called expressed, meaning a particular gene, if it is producing a lot of copies of RNA, the amount of expression is proportional to the amount of copy of that RNA molecule being produced. Highly expressed means high amount of that particular RNA molecule is produced. So the most recent advent or the current state of the art is something called as RNA sequencing. This principle works based on, you again use the same methodology, how do you prepare the samples for your microarray, but instead of using the existing set of molecules and then you just try to use light intensity to understand the amount of gene expression, what you do here is you basically sequence meaning you capture the readouts, what is the alignment of each RNA molecule. So it's something like, as you see, you just start to write the sequences. Something like how they do in the orchestra. Like as they see, they start to play the different equipments, right? So as it is, the computer machine, what it will do, it will read out the sequence of different RNA molecules and it will capture that in a digitally recorded file, which can be analyzed later. So in overview, this is how this happens. The entire mRNA is first taken from any of the sample, but this is unstable as I mentioned earlier. So we fragment it and then we couple it with another template so that we make a DNA molecule. Because this is a complementary to an existing RNA, we call this as cDNA, complementary DNA molecule. And then we generate multiple copies of this using a polymerase chain reaction. So I will not go into those technical details. You are basically amplifying the number of copies available, and then you put into a sequencing machine and you try to record the sequence in a digital format. So this is how it will look like basically. Okay, so once you have this format digitally recorded, what can we do with this? So this, from here on, the entire focus of this workshop would be on the data analysis part, so which would be of interest for you as well. So what you get from the machine is something called as raw reads. But this may have some errors, the machine errors, or even the sample preparation errors and so on. So initially we need to do a quality control, meaning like whether the sequence has captured enough amount of RNA molecules in the desired quality. So once it passes through that, and we also use something called as adapters, which is basically for each RNA, for the machine to detect that it is an RNA molecule, you add something in front of it. So that once that sequence is followed by something, the machine will detect this is an RNA molecule. So that part is called, that's a synthetic part, that is called as adapters. So you have to also remove those so that you don't need to analyze those. Once you are able to do that, what we do next is the different genetic materials, different genes, and what the sequence of that, the exact sequence of each of those genes is already known for many of the organisms. That's what we call it as genome. So it's usually assembled. So you sequence all the genetic material and you nicely assemble something, and that is actually called your genome assembly. From your RNA data, this RNA sequencing data, what I try to do is, I try to align these red reads are what I extracted, and I try to place it next to the genome assembly. And then I identify, oh, this read is corresponding to this gene. This read probably corresponds to this gene. This read corresponds to this gene, and so on. So that's how I detect what are the genes actually being expressed, which are the RNA molecules of those genes are produced. But this is still, you remember, in read format. So for example, for a particular gene, you may have several reads. For another gene, you may have slightly lesser number of reads. For a different gene, you may have an intermediary number of reads and so on. This is difficult to handle, right? So then we try to change this information depending on the number of reads extracted into a numerical count, which is what we call it as gene expression counts. So that process is called as counting, and this is a numerical value. So for example, particular gene across samples, you will have values like 10, 20, 30, which means this gene is expressed 10 times in this, 20 times in this sample, 30 times in this sample, and so on. So from this information, you may then do differential expression analysis, meaning like whether that particular gene is really differentially expressed between the two type of comparison or more than two type of comparison. And if it is really differentially expressed, what does that mean? Is it linked to the observed variation? See, you may have hundreds of genes which are different between a disease and expressed in a different amounts between a disease and a normal condition. But do they all contribute to the outcome of a disease? That's not actually sure. So in order to do that, we may do something called as enrichment analysis or a biological interpretation, which we will also go through. So again, I want to reiterate. So in between this differential expression from the counts, you can also do a unsupervised analysis. I guess you may be familiar with what is supervised and unsupervised analysis. Are you familiar with that? Yeah, so unsupervised analysis, yes. Unsupervised analysis meaning you would want to see if you have different number of samples, say 4 or 5 different type of samples, you may want to see trends, whether any two samples are similar than other two samples or other sample. So that reproducibility and similarity across different samples can be analyzed using a dimensionality reduction or unsupervised analysis. So this is how a sample data will look like. So here I have taken information which produces a cell line from an animal cell, which is producing a particular protein which is foreign to that. It is not actually having a gene to produce that protein, but we have inserted it, and that is what we call it as producer. And this is a non-producer which does not produce that particular gene. Because we are inserting a foreign gene into that organism, it may undergo some genetic changes. So that's what we are trying to analyze here. We are going to go through whether the gene expression of how many of the different genes are, whether it is different between the producer and non-producer. So if you look at here, we have taken 3 replicates, meaning in the producer category, I have taken 3 individual cells for the profiling, or 3 group of cells, you can call it however you want. And then I've tracked the expression of different genes. So this data is what we get it after the counting procedure. And similarly, we also obtained it from the non-producer, where again we take 3 samples as for the processing. Okay, so with this, how can we do something called as unsupervised analysis or dimensionality reduction? So I will want to shift my focus a little bit. I will explain more in detail about dimensionality reduction, what it is, and why we do that for a gene expression data analysis, and how we can make sense of that data. So dimensionality reduction is nothing but a technique which will be used to reduce the number of features. So in machine learning lingo, you may be already familiar that features are nothing but, in this case, the different number of genes. So typically when you are handling a animal cell, you will be handling around 20,000 odd genes. So this is a large number of features to handle, and not necessarily all the features are informative. Meaning, like, there won't be a variation in the expression of all the 20,000 genes when you look at different conditions. So you may want to look at only the relevant ones. So that is what we are going to do with dimensionality reduction. So why we do dimensionality reduction? With the raw data, you will have an increased computational time as well as resources. High amount of RAM is required and so on. And also in visualization, you may have difficulties. So that's why we want to remove the redundant or irrelevant features so that we can make our data more manageable. So let's take an example. So I have a data set where I have monitored a particular gene's expression in 6 different tissues. So I have taken the same gene's expression values from a lung tissue, stomach tissue, kidney, heart, pancreas, and thyroid. So if I just have one particular gene expression value, in terms of visualization, if I just use the expression as the y-axis, then I just see a kind of clustering that in the lung, stomach, and kidney, the expression levels are similar, whereas it's very high in heart tissue, whereas the pancreas and thyroid, it's somewhere in the intermediate level. This is very simple because I just have one gene. What if we have 2 genes? I can take 2 dimensions, gene 1's expression in this dimension, gene 2 expression in this dimension, and I can still see some patterns. So there are 3 genes in 3 tissues which cluster together, 2 other tissues which cluster in a separate manner, and 1 is complete outlier, which is the heart. But this is simple because we just have 2 dimensions. Imagine you have 20,000 genes. You cannot create a plot to visualize how they are different, and that's where dimensionality reduction is useful. So I will be going through in detail a particular method which may be very useful, is called as principal component analysis. This is a very commonly used dimensionality reduction technique. So here what we do is, this is a plot which shows the entire data set after the dimensionality reduction. I am able to see that the transformed data is able to separate my heart tissue far away from pancreas and thyroid, whereas I see that there is a similarity in lung, kidney, and spleen. So how we have done this? Let's see. So the first step in doing any principal component analysis is you take the individual values in each dimensions and then you try to center it or scale it, meaning that so here you have the expression over a certain range, right? And we have to take the values and transform it towards the average value of that particular gene. So that's what I'm trying to do here. So there is a value in 10, 15, two values near 15, and one near 45, and two near 25. So I have to take the average, which comes somewhere around here. And after I transform this into the mean of the particular dimensions, then what I need to do is, I need to draw a line which passes through the origin. And it also maps to the mapped individual data points, which is on the lower dimension. And we need to calculate, or we need to rotate this line. So we need to rotate this particular line such that the distance between the line to this point is minimized. So in this way, currently it's already minimized. So if it is slightly on this direction, then the distance to this. So we have to use the sum of all these distances so that it's exactly minimum at a particular point. So it has to pass through this origin as well as to reduce it. So here again, the thing which I would like to highlight is we have just used 2 dimensions, but in reality PCA will work with several dimensions. So it will try to pass through for all the perpendicular, all the dimensions in a perpendicular manner. And the new plane what we are going to draw will also be perpendicular to all those dimensions. And this transformed plane is actually called as principal components, which satisfies 2 things. One is it passes through the origin. The other one is it has the minimum distance from the individual points to the point of intersection. So if you look at the point of intersection, it's nothing but it's following the Pythagoras theorem. So you have the origin here, the data point here. The distance between this is fixed. So as long as you are rotating your PC, A and B varies. So if you are trying to minimize A, you are trying to maximize B. Or in other words, you're trying to minimize B, you're trying to maximize A. But our ultimate aim is to minimize this A, which is the distance from this point to this one. So in other words, you are trying to maximize the distance from here to here. So that's what in computationally all your algorithms will try to do. The amount of variance which is captured in that particular principal component. So if you use that logic, your first principal component will have the maximum amount of variation. So then you may draw another principal component which is exactly perpendicular to this as well as to others. So then it will have the second least variation. So the first one usually will have the maximum amount of variation. The second one will also have the second maximum variation. So we calculate the maximum variation using Euclidean distance. So I calculate the distance from the origin to each of these points. So I use like D1, D2, D3 and so on. And then I just calculate the sum of all these things. So the sum of squared distances will tell me whichever will have the maximum will be my PC1 and the second maximum will be my PC2 and so on. And from this we can also have we can also identify how much each of the genes or each of the dimensions is contributing to your variation. So in this case, your slope is provided by say 3/2. So on this axis, I have my gene 1. This axis, I have my gene 2. So which means the contribution in terms of this axis or this gene is lesser when compared to this gene. So that's why gene 2 has more in terms of that particular principal component than gene 1. So this will be slightly tougher to follow, but with the practical example, it may be slightly become easier to follow. So when we know the individual variation captured, when we take the fraction, and then when we multiply it, we will be able to call something like the total variation captured in terms of percentages. So in any principal component analysis, you will see in the axis is PC1, within the brackets some amount of percentage, which is nothing but the variation captured in that dimension. Usually you will see a 2-dimensional plot or a 3-dimensional plot, where the first two PCs will capture more than say 70-80% of the variation, or in certain cases, if it is not able to capture enough variation, you may go up to the 3rd dimension. Okay, let's go through a practical example. So I'm taking a pause from my gene expression data. Let me give you a real life scenario to explain PCA more in detail. Then with the data set what I showed, we will be going through a actual demo through this particular workshop. So in this, I have a data set where different athletes who performed in different events in a sports event, and they have scored certain points. It may be difficult to analyze who is better in which of this. So I have to eyeball differently, like I have to plot all these things and I have to look at all these things. The double slash means there are a lot of readings in between. Similarly, there are also several athletes. So this is where your PCA can be really handy to see whether certain athletes are able to perform very well in say certain amount of events, or certain events, is it distinct from other events? When individuals are performing well in a particular event, they are not at all performing well in other events. So you may have to, you may be able to get these kind of patterns very clearly from a PCA. So once we do the PCA, the first thing we should actually look into is the amount of variation captured in each of the PCs. So here we are able to see that the first PC is able to capture around 40%, the second PC around 18%, 12%, and so on. So usually when you sum it all up, it will be 100%. And the number of dimensions or the number of principal components you get is actually based on number of features or number of samples, whichever is the lower. Most of the cases, your samples will be lower. So if you are dealing with, say, in this case, say 10 events, 100 athletes, then the dimensions would be 10 principal components. So then I plotted the principal component 1 and principal component 2, and I see different grouping of events. So this suggests that those who are there is a similarity in 100 meter, 400 meter, and 110 meter hurdle. People who are performing, they are able to perform very similar in all these three things. Whereas there is a diagonally opposite characteristic is observed in javelin, long jump, shot put, and high jump. So this shows that those who are able to excel in this, they may be performing very low on this. And those who are performing well on this, they may be performing very low on this. And you also have a different category, which is long distance running, high 1500 meter, and also pole vault, which is somewhere a different category. So from this, I could able to see that the data is actually clustering into 3 different clusters. I could see a cluster here, I could see a cluster here, I could see a cluster here. So this will be the practical usage of PCA in a gene expression analysis. So when you do a PCA of the example what I showed, that's why we were able to see the expression of 3 tissues closer to each other, 1 tissue far away from others, and 2 other tissues somewhere closer. So this tells us, in terms of just based on gene expression patterns, they are very dissimilar. 3 are similar, but it's very dissimilar from the heart. So another, yes.
This one, you can get it very easily in pandas CORR, right?
Pandas CORR.
Python.
Hmm?
There is a correlation we can create. We have any number of features.
Right.
So it will create that n by n matrix.
Yes, right. So what you are talking about is correlation between any 2 variables. So, but you have to analyze it between 2 variables at a time. So it will create 2 by, I mean, like it's a matrix, right? So you have to create any 2 variables, you will be able to see the analysis, I mean, variation at a time.
N number of variables, n by n, it will show a matrix with the color coded.
In each plot, you may be able to see only the variation between 2 variables, I guess.
At a time.
Yeah, at a time. But what PCA can do is it works with something called as covariance, meaning that it will be able to simultaneously look at the variation in all the other variables. So that's why we call this as a multivariate analysis. What you are suggesting is a bivariate analysis. So at a given time, it will be able to see the variation between any 2 variables. But this can look at all the different variables together. So this is, you will get the overall trends, not just, I have to do that, I have to create it so many times for any pair, whereas here I will be able to get all the trends at a single group. So that's the major difference. So on the right hand side, what we are seeing here is the contribution of individual athletes to the different events. So from this, what we can say is that the grouping what we observed in 400 meter, 110 meters and 100 meters is mainly because of the contributions by these athletes, Bernard, Henro, Yurko and so on. Whereas all these athletes, they performed so equally in most of these events that they are not able to excel in any of this. So that's why they are able to group here. Whereas those who are grouped here, they did very well in these, say for example, Clay and Serbel, they performed very well in long jump, but probably very low on 100 meters and so on. So this is called as biplot, where in the gene expression data set, if you want to do this, you may be able to see you saw that the heart clustered far away from others, right? So you may be able to see a set of genes around that particular heart tissue, which will tell you why heart is different from the other tissues because of the expression of those genes. That's why you are able to see that as a separate cluster. Okay, so once you are able to do this, so what we will be also going through today is, we will also check how many of this differential, how many of these genes are differentially expressed between your control and the diseased condition. Say if you have, usually gene expression data analysis is done between two sets of samples. So in that case, you may want to do a hypothesis testing to see whether your gene is really differentially expressed with the statistical confidence. Usually hypothesis testing, we do it with students t-test because you know that data may be normally distributed. Normal distribution is nothing but when you have hundreds of measurements, most of the measurements would be following a Gaussian distribution or a normal distribution, where it will fall towards the mean of the data or around the mean of the data. But in gene expression analysis, that won't be the case. So you may not be able to follow a student's t-test. So we usually follow something called as Poisson distribution or a binomial distribution to do the hypothesis testing. So another thing which we need to note in gene expression analysis is, since we are going to do this hypothesis testing for all the genes separately, so imagine you are having 20,000 genes in human. So you are going to do 20,000 hypothesis testing. So there may be a simple chance that some of these tests could result as a positive purely because of a chance. So there could be a false positives or true negatives as well. So in order to mitigate this, what we do is we also do something called as hypothesis correction. So we also adjust our p-value such that we account for the errors possibly what we can encounter in our hypothesis testing. So once you are able to do this differential expression analysis, what we can do is you will get a set of genes which is differentially expressed between your normal and the diseased condition. But it does not say anything, right? It just gives you a list of genes. Then what you can do is something called as enrichment analysis, which can be of 3 types. So what we are going to demonstrate today is something on the over-representation category, meaning that from the list of genes what you have, you will see from each pathway. So how it works in biology is each gene is probably mapped to a particular pathway. So a pathway may have 100 genes, another path may have 10 genes, another pathway may have 50 genes. So out of this 100, if there are 40 genes present in your differential expression analysis, so it is over-representing that particular pathway among all your differential expression genes. So that's what we try to see. So that means among the differentially expressed genes, the genes are specifically tilting towards a particular pathway. The collection of genes from a particular pathway is differentially regulated, so which may cause a particular outcome in your disease. So you try to also then look into the earlier studies which has hypothesized, okay, this particular pathway has a role in this disease. So then it will strengthen your further understanding about the gene expression analysis. Then you can also do something called as functional class scoring, where you can just try to group genes based on similar expression patterns. You do clustering. All the genes which have a similar expression patterns, you may try to group them and see whether they belong to a particular pathway or not. The last one is you may try to do a network-based analysis. So the next door, my colleague Karthik is doing workshop on network analysis, which can be also applied for genes, where you may try to see different genes, whether they are connected through common pathways. Some cases, genes may be classified in multiple pathways. So you may see the differential expression, not only in a single pathway, but also in a connected different pathways as well. So this will give you an idea how each pathway is affecting the other pathways through a network connections, through that association between different genes and the pathways. Okay, so we will be actually getting on with the workshop, where the TAs will be performing step by step what I've gone through now, and then you can be able to follow that. But before that, please ask me if you have any questions. I will be able to clarify that. Any questions? Yes.
So all genes like determine, or we have a data for that. Whatever genes are there.
Yes. So that is what we call it as the genome assembly, meaning like the genetic material of humans from multiple humans is taken, and we know what is the different genes present in each human. So but the amount of each gene present, that may vary at the gene level itself, and expressing, which is the copy of that becoming RNA, may change in liver tissue to heart tissue to brain, but genetically the cell is identical in a given human. So it will be same. It's just the gene expression which changes across different tissues. Okay, so if we don't have more questions, I will be still around. I will be able to answer. So let me introduce my TAs, Vikash and Deepika. So they will be taking over and they will go through this. So I'll hand it over to Vikash.
Good afternoon all. So we have our sample data in this QR, and you have the URL as well. You can just scan it or type it down and just access the Google Drive folder. So I hope every one of you can access the particular folder, and you will be having the data files as well as the Python script in it. And the next few minutes we'll be discussing about the pre-processing of the data, followed by the principal component analysis. So for this analysis, we'll be using Python. In specific, we'll be using the scikit-learn library, And scikit-learn library has its own PCA package, which has default functions in it for this analysis. So initially we'll be starting with importing the required packages for this analysis, which would be NumPy, Pandas, followed by matplot library for plotting the graphs and things. And we have Seaborn as well for a very good representation of the graphs. And we have the scikit-learn library for PC analysis, and before starting the analysis, we'll need to scale the data to a smaller scale so that we don't get lost with the variabilities in this data. So once you are done with importing this libraries into your script, the next step would be importing the data into the script. So for that, we'll be using the pandas function called pd.read CSV. And in the particular folder which I shared, there will be a data set called tissue_datasets. So that would be the data which we'll be using for this PC analysis. So we can import the data into the script using this option, upload session storage. So once it's successfully uploaded, you can see the particular file in the left window. So I'll be importing this particular data set into Python, and I'll be storing it as a variable as data. And this is the command to just pick the specific index of a particular data set, where I'll be taking the first column of this data as genes, which we'll be using in the further steps of the analysis. So it would be data.ilog of this colon represents that you'll be selecting all the rows in this particular data set, and 0 represents I'll be choosing the 0th column of this data set. And data.head would give you a summary of the data set rather giving the complete data in it. So we'll be seeing just the first 5 rows of this particular data set. And as you can see, you have genes in the first column, followed by you have 8 different samples in it, in which you have 2 kidney samples, 2 liver samples, 2 lung samples, and 2 spleen samples. So for an ease of understanding, I'm trying to just rearrange these columns so that you have the kidney samples and each organ samples in a consecutive way. So you have kidney samples consecutively followed by the liver samples, lung samples, and the spleen samples. So for that, you'll be using this particular command, which is I'm storing this particular data set as data_df. So where you'll be choosing the columns with respect to the column names. So in particular data, my first column would be genes, followed by the kidney, followed by kidney_1, 2. And in that way, I'll be arranging the columns of my choice. And again, for a presentation, I'm using the head function to visualize the data set. And in Python, we'll be calling this as data frames. So as you can see, the scale of variability is too much. So you have kidney samples, the first gene, you have kidney sample has 1279, Whereas if you have in liver, it's just 26. So if you can visualize, it's like that variability is so much. So in your analysis, you'll be getting some irrelevant interpretations or something. So in that case, you'll be trying to scale down this data set into a smaller scale. For that, we'll be using a method called mean centering, where the data would be scaled down to a smaller scale with respect to the mean of the particular sample. So in that particular data set, I'll be taking each row, find the mean of it, and I'll be subtracting that particular mean from each data points divided by the mean. So in that way, I'll be getting a mean centered data points. So this is the just a mathematical representation of that operation. And I'm going to store this mean centered data in the variable name scale data. So you can visualize that certain genes would have a count of 0, and just think of the mean of that particular row would be again 0. So 0 minus 0 divided by 0. In that case, you will be getting an infinity. And in programming language, this infinity will be represented as NaN values, which is not a number. With this values, it's not possible to do any analysis out of it. So in that case, you have to do some manipulation so that you don't get any infinity values in your process data set. For that, the first step we'll be doing is, we'll be adding 1 to each data points, and then we'll be processing this data. So now we'll be having 1 as a mean of that particular 0th row, and if you take a mean centering of that, then you'll be getting 0 in your process data. Now it's possible to do some analysis out of that. So that's the first step we are doing. We are taking the all the rows and the first column to the last column. So this represents that you are taking columns from 1 to the end, and we'll be adding NP is a numpy array of ones, which is of equivalent to the shape of this data set which you are taking, and that would be stored as scale data. And the next step, you are calculating the mean of each column. So you can use the function called mean, where it calculates mean with respect to the rows. So axis 1 represents the column, whereas axis equal to 0 would represent rows. This may sound confusing, but the thing what you are trying to do is, you are taking column values of each row and you will be performing the mean calculation. So now we have the scale data, followed by you have the mean, and using a for loop, I am trying to pick a particular row, perform the same operation out of it, and I will be updating this scale data with that mean centered data. And at the end, I will be just, for an appealing view, I am just adding the gene list in the first column, followed by the mean centered data. Now we can see that the variability is somewhat reduced. So it's hardly in within -1 to +1, I guess. So it's better to do this scaling. So right now the scale is reduced, and now it's possible to do a better analysis out of it. And for a better understanding of the scaling, I have performed a box plot of it. So where you can see before scaling, you have a lot of outliers which are like too much away from the actual data sets, whereas following a scaling, it's somewhat reduced. And the sample 1 to 8 represents the each tissues. So with that, our data processing has been done, and right now we are ready to start off with the PC analysis. So for this PC analysis, first step would be, we'll be transposing this data such that we'll be having the tissues in the rows followed by the genes in the columns. So as sir has explained, your tissues are like the samples and your genes are the features. So in this particular data set, we have 8 samples, 21,488 features. So the first command is for the just transposing your data, and followed by you are just naming the columns, and you just give the names to the tissues. So with this particular data matrix, we can start with the PCA analysis. So first, I am just picking the numerical data of this particular data frame and storing it as sample underscore df. The next step is initializing the PCA class. So PCA is the scikit-learn has its own PCA package in it. So initial step would be initializing those package into the script, and we are trying to fit the data into this PCA function. So that's what we are trying to do with the PCA.fit. And the first step would be finding out the projection of all the data points on different principal components, which are like so most likely the number of principal components would be equal to the number of samples which you are taking. So the command in PC is that principal.components would give you the projections of each data points on different PCs. So that you can visualize through this particular image. We have the raw data set plotted in the axis, and you are just projecting it to the first principal component, followed by the second principal component and the third principal component. And you can see from the image itself that we will be having a very good separation in data points, projections in PC1, whereas in PC2 it's somewhat reduced. You can see there's a overlapping in this data points, whereas in PC3 it's relatively very less. So we have too much of overlap in this projections. So PCA component would give you a matrix which is equivalent to the number of, where your number of rows would be corresponding to the number of principal components or the samples, and the columns will be equivalent to the number of features in the data set. So we will be having projections of each data point on different principal components. So because of this, the view has been truncated here, whereas you will be having 8 different rows here, followed by 21,488 columns. So next step would be finding out the singular values for this matrix. So basically singular values are nothing but the square root of eigenvalues of this particular data set, like positive eigenvalues. So those are called the singular values, and basically your singular value represents the kind of a weightage of each principal components. So it would be arranged in a hierarchical way, where your principal component 1 would have the highest weightage, followed by the rest of the principal components. And this would be equivalent to the explained variance as well. So your PCA.explained variance would give you the explained variance percentage of each principal components. Again, we'll be getting 8 different values for that. For a better visualization, we'll be plotting a bar graph of this explained variance, and you can see that your principal component 1 is able to explain 31% of the variance of this data point, followed by the principal component 2, which can explain 27.4% of the principal variations. So now we'll be trying to reduce the dimensionality of our data using a function called pca.transform of the particular sample data df. And for plotting purpose, we'll be creating a data frame with that particular transform data, where you'll be having your principal columns as principal components, which would be 8 cross 8 matrix. And for understanding, we have included a tissue label as well. So in other words, we call this as PCA scorings. And next, we are trying to plot this in the form of a plot. So we have plotted principal component 1 as its principal component 2, and you have the amount, percentage of explained variance of each particular principal components. And on plotting, you can see that your liver sample gets clustered together and your kidney samples clusters together. And in a separate way, you have lung samples represented by green points, and the red points indicates the spleen. And also you can understand that your cluster of kidney and liver is somewhat close together, which means they have a positive relationship between them. And to understand the correlation between these different tissue samples, we just plot a correlation circle for that. And you can see that kidney and liver have a positive correlation, whereas your lung has a negative correlation with respect to the other 3 samples. Similar way, spleen has different correlation with respect to the other 3 samples. So your basic interpretation of this particular plot is that you may understand kidney and liver are some organs which perform the detoxification in this human or any organism's body. So that could be one reason why they have a positive correlation between themselves, whereas lung and spleen are somewhat different from these 2 organs. So that could be a primary interpretation of this data. For a better understanding of these correlations, we go for further analysis like the flux and functional enrichment analysis, which should be handled by Deepika. Yeah.
When we started with the topic that we want to make a group of genes.
Yes.
Now in this chart, I can see the organs, so I am confused.
Yeah, based on the genes, the organs has been clustered as separate. Gene expressions.
So what is our purpose here?
Based on the gene expression, we are trying to cluster these organs separately.
So here, you're trying to deal with 4 different issues. And you want to understand what is the difference between the 4 issues just based on gene expression. But subsequently, we will also show See, you see that. Which two tissues? Okay, so if you want to understand the difference between spleen and lung, you may do a differential expression. This is pairless, like lean and splunk, or like lung and spleen, or spleen and liver, kidney and liver, and so on. Then you will have a list of genes which is differentiated, from which you can do functional analysis. What she has shown, what he has shown now is when you have more than two samples, what is the overall interpretation you can get from that particular?
So now we got here that this organs are similar.
This two is similar when compared to the others. That is the main take-home message. And there could be a cluster of genes which contribute to the similarity in liver and kidney. And there could be a cluster of genes which is causing the dissimilarity in spleen or dissimilarity, which you can take from the loading slot, which he has not covered because of the time. But you can get that information. Any other questions?
Okay, so is it okay? So good afternoon all. So after plotting the PCA to understand the overall variance and also the batch corrections in RNA-seq, we'll be moving forward with the differential expression analysis. So right now we'll be using a function called PyDseq2 using a Python implementation. So PyDseq2 also normalizes the data, estimates the variance of the gene, and also identifies the differentially expressed genes across different experimental conditions by using statistical measures for providing robust and specific interest in the expression of genes. So first we'll install PyDseq2, Matplotlib, and also Seaborn. So PyDseq2 performs the differential expression analysis, and Matplotlib is for visualizing and plotting the data, and Seaborn is for enhancing the statistical test. So I think you all are working towards this. So after you install it, then you're going to import the libraries. So as said before, you're going to import pandas, Matplotlib, Seaborn, and also NumPy to do the visualization, manipulation of the data, and also for enhancing statistical tests. The next is going to be the loading of the data. So as said by my teammate Vikas, we can either read the data from our drive, or you can upload it in the folder using upload session. Either way works. So first we are trying to create a data frame where we're trying to read the tissue dataset.csv, and for doing differential expression analysis we need two data sets. One is a count data and the other one is a metadata which has the information about the samples present in the count data. So we are trying to read that in tissue_dataset into the data frame, and tissue_metadata into the data frame 1. Then in a normal way, Python takes the dataset as the first one as 1, 2, 3, 4, and the second column is the features, and the next one are the samples. So we don't want our features to be a regular column. We want it to be indexed as a row label for us to perform the further analysis. So we're trying to index our feature column, and I'm trying to name it here as gene. Then we are transposing the data. So the pre-processing step is done here. Then, as I said before, we need a metadata to perform differential expression analysis. So here in the metadata, we have information such as cell, what type of sample it is, if it's a producer or a non-producer, what kind of batch it belongs to. So I'm trying to take my cell as a group, and based on what condition it's going to find the differential expressed genes, it's based on batch. So because that's the two information I'm having in my metadata. So I'm renaming the cell and the batch as group and condition in my metadata, and I'm again indexing my column as the indexing my column as the row names. Then after this, as said before, we are having a library called PyDseq2, which performs the function of differential expression analysis in Python. So we are first importing a dseq data set. Then we are trying to set a default inference, as in like we are trying to give 8 CPU for this particular job. Then we are importing the data set. So as we can see here, inference equal to default inference n underscore CPUs. We are giving 8 CPUs for this particular job to run. Then we are creating a data frame which has count data, the metadata, and also the design factor, which is the condition which we are going to do the differential expression analysis, and the inference. Next we are trying to do, as said before by Sir, we are trying to do one second. So we are trying to do a statistical test to understand how the data is being produced. So here we'll be doing a Walsh test to do So here we'll be using the Walsh test to do the false discovery rate to remove the false discoveries found in the data. So the stat underscore res is a variable under which the dseq stats is being done, and the summary of the data is shown below. And we are taking a variable called df under tmp to store the results of the statistical test we've got. So as you can see here, we are fitting the size factor first, and then the dispersion to estimate the variances in particular gene, then mapping the dispersion, then running the val test, as I've said before, and the log2 fold change and the val test p value is shown between different conditions. That is the batch 11 and 12, which I'm focusing on. Then, so this is the result we've got. So the genes and the respective base mean, log fold change, and p value. So these genes are the differentially expressed genes. So I'm doing a filtering process here where I'm trying to remove p-values where the genes have p-value 0 and p-value less than 0.05. And I'm keeping the p-values less than 0.05 because it's statistically significant. And I'm putting it into a variable called df_temp_filter. And I'm writing the file as to a variable called result_filter.csv. So once we are done with this, then I'm trying to find if it's a upregulated or downregulated gene list. So I'm using log2 fold change and negative logarithm of p-value to understand the magnitude of change in the gene expression and also the significance of the log fold change. So for that, we are going to use a plot which is called as a volcano plot to visualizing the genes. So here we are going to do a scatter plot to visualize the volcano plot. So SNS scatter plot is a function in Python where it uses x as the log2 fold change, which is a change in the magnitude of the genes, and y is the p-value, which is the log10 p-value, and the data frame, and it takes in the regulation to see if it's an upregulation or a downregulation. And the palette is basically the color palette. If it's up, it's going to be red, and if it's down, it's going to be blue. Then the title of the plot is volcano. Then the x and y labels are marked here. So the x will be log2 fold change, as I said before, and y will be minus log10 p-value. Then the plot AXH line is a horizontal line. So we are trying to plot the horizontal line to find out the log change. So the log2 fold change. So points are the points as in the features. The genes above 0.05 are statistically significant. And then the plot of AXV line is a vertical line. So here we are trying to draw two vertical lines, one for upregulation and another for downregulation. So then we are creating the volcano plot and using the df_temp, which is the variable in which we stored our gene list, the differential express gene list. And then we're saving the up and down regulated genes and we're writing it to a CSV file. And as you can see here, this is the plot that we generated. So the points in red are upregulated and the points in blue are downregulated. As you can see, the points here, the log10 p-values shows the statistical significance of the fold chain. And yeah, that's it. So after this, we have a gene list. I've stored it in the folder. Yeah.
So I would like to explain few things more clearly. So as you see here, she has done a differential expression analysis between two tissues. So 1,2 you may assume as spleen, 1,1 you may assume as lung. So from this, what you get is the output of each of the gene, whether when compared to lung, what is the amount of fold change in the expression in spleen. So that's what you see it here. So it's a log2 fold change, meaning that the value, the fold change is calculated in a log scale. So minus 3.7 is roughly a very high fold change. It's very highly expressed in 1,2, which is probably your lung, than your spleen. And for that corresponding, you have a p-value as well and the adjusted p-value. So if it is something very significant, because imagine you have two samples, right, for each of them. If it is statistically significant, you see the same pattern between the two samples. Like if it is lowly expressed in both the samples when compared to the other sample, then you will see that the p-value also very high, significant, which is 0.05 or much lower than that. So from this, it may be difficult to interpret. So that's why we use this. We use this volcano plot. What it suggests is all your 20,000 odd genes is here, but some of the genes, they are statistically significant, which is in this quadrant above the value of this cutoff. And if you see the direction towards the lung or towards the spleen, these genes make sure that this is more as a spleen. And these genes, they make sure that they are very highly expressed in your lung. So that is the advantage of doing a volcano plot. So your differentially expressed genes are a set of these genes and these genes. So you may call this as upregulated in spleen and downregulated in spleen, or upregulated in lung and downregulated in lung. So that's what we call it as up and down regulation. So taking this, she may do some enrichment analysis to see what are the pathways which are different between spleen and lung.
So there is a standalone tool for doing the enrichment analysis. It's called David, which is nothing but database for annotation, visualization, and integrated. So this is the home page of David. So we'll click on the start analysis to proceed with the So I'll be taking the first column, which is the gene, which is the features, which we'll be using in the David. I'm going to paste it in the upload section. So before running the analysis, we need to select what kind of the identifier our gene list is belonging to. So we have official gene symbol as our identifier for the gene list we've got. So we are going to select that in the drop down menu. And since our tissues belongs to Chinese hamster ovary cells, which is a hamster, so we'll be choosing the species which we are going to do the enrichment on, and the scientific name is Chrysicillus. One second. Grisis, which is the scientific name of Chinese hamster ovary cells. And then we are going to see what kind of list type we need, if it's a gene list or a background we need about the particular data. And then we are going to submit the list. The next is the analysis using the David tool. So here we have functional analysis. So we are going to select the functional annotation tool. So we'll get a summary of what are the different functional annotation, gene ontology, the pathways which we've got in the screen. So since we wanted to see into the pathway analysis, we are going to click the drop down menu and select the get pathway analysis. So as you can see, we are getting 30 hits for the respective genes. We have pasted. So there are a lot of options you can explore if you want a fold enrichment analysis, if you want to do Bonferroni correction or Benjamini correction, you can use that. So with this pathway list, you can do a visualization to see how much enrichment is done for each gene in a particular pathway. So there are independent website or tools you can use, or you can use Python Seaborn to visualize the data. So as shown here, this is the data that that is being visualized, which I've shown right now. So I did a visualization plot using an independent plot, and as you can see that systemic lupus erythemus is a particular pathway that is being enriched highly. Next is alcoholism, and the next one is neurotrophil extracellular trap inflammation, and you can read about it in the Google, I guess. And that's all. Thank you.
So here, this analysis, what it suggests is, so when you see this, when you see these pathways, so this suggests that between the comparison what we did, lung and spleen, these are the pathways which are kind of have the genes which is differentially expressed or causing the difference between how a lung should be or how a spleen should be. So that is why you are seeing a set of pathways which are highly enriched. So they have a very high p-value, very significant p-value, and a large number of genes collected in that particular pathway as well. So from this, you may get an idea what kind of pathways are differentially regulated in your two tissues. So in this particular case. So you may also see that between a diseased case versus a control condition, normal population and so on. So this is how a normal gene expression data set works. So I hope we have given a taste of how a gene expression analysis can be done using the RNA-seq data. But if you have any questions, please let us know so that we will be able to help you with.
Can you give some broad idea that how the same thing we can use for, let's say, normal patient versus a patient with, let's say, some type of cancer?
So you can do the exactly same analysis. So there you will pick up certain